Devendra Bhaskar is with you, and Miss Tanya Samrat. So she'll be here and assisting you. And after a while, Dr. Geetika Sethi, she will also be joining. She will give you one very interesting lecture. It will be basically on a very important platform called R. It's a R language and R studio that will be very useful for analyzing this large voluminous data. So that will be one. So with this, if do we have Dr. Harpreet Singh online?
He's trying.
Yeah. अभी तक हमारी आवाज उधर गई
अब जा रही
Good. Thank you. So little bit hiccups, but still we are online.
Yeah.
So along with this workshop, I think we have given you some of the workshop material which was coming from Dr. Pradhan's side. So you will find that there will be a something this kind of page. Can you see this page? Just behind your program page. So there is one QR code is there. In case your phone is not able to scan this QR code. So can you just find out if this QR code is working or not? So you can scan it. So what do you find in that QR code or that shared folder? There is a program there. Is it there? And then there is a list of participants. It's very important that you know each other when you are working here. And then literature on NGS data analysis. So this major part coming from the ICMR Ames Genome Center. Okay. So in the course of time, you may be needing these kind of that basic knowledge. You can actually have a quick read in case when we have some pause or some coffee time. So at least you can have a quick read, and if you have any question, you can talk to the resource person. Is Dr. Sab there? He is busy. Okay, so we'll continue. I think in between somewhere, if we get a chance, so we will have him on for 5 minutes on the line, and he'll be interacting with you. And I'll give the dais to Dr. Pradhan from here. But before we actually start this, I think you have already got introduced to our resource persons. And I will ask right from this side, the students can introduce themselves. You can give one mic to them. Okay, and other students ko bata do, so that they will have clear idea. You have to tell your name and your affiliation. In case you are from department, you can say that you are doing a specialization in genomics and genomes and evolution, so that they will have a clear idea. Resource person will have an understanding what background students are there, so that they will be accordingly going into the depth of the subject.
Very good morning to all. My name is Rohan Dahel, and I am pursuing my master's in zoology from the Department of Zoology, University of Delhi. I have an inclination towards fish biology, but I think that this particular workshop will be very beneficial for me, as these practical applications are useful in every field these days, in the major field of biology. So I'm looking forward to learn a lot today.
Good.
Good morning, everyone. My name is Harsheen, and I'm also pursuing masters from the Department of Zoology at University. And I am currently specializing in fish biology. But I think that since data is everywhere and data analysis is a very important part of research nowadays, so it will be very fruitful to be a part of this workshop. Thank you. Good morning, everyone. I'm and I'm pursuing my master's from the department itself. And I'm a final year student, and my specialization is in genome and evolution. So this is basically part of my specialization. Good morning. My name is Devanshi, and I'm doing masters in zoology from the department itself, and I'm specializing in genomes and evolution. Good morning, everybody. I'm Dr. Sonia. I'm from MR's Radio. I'm basically a pharmacologist. Although we cannot separate ourselves from data analysis, myself, and it's very important and integral part of the science today. So I'm looking forward to learn quite a bit. Thank you. Good morning, all. I'm Priyanka Sani. I'm pursuing my master's in zoology from this department itself, and I'm doing a specialization in genomics and evolution. Thank you. Good morning, everyone. My name is Shen Qureshi. I'm pursuing my specialization in genomics and evolution, and I'm looking forward to learn more about it. Good morning, all. I'm Vanita Kumar, and I'm also pursuing master's in zoology from the Department of Zoology itself. And I'm doing specialization in genomics and evolution. And I'm also really excited and looking forward to learn more about the data analysis. Good morning, everyone. My name is Rita Marik, and I am pursuing a master's from Department of Zoology, and my specialization is genomics and evolution. And I'm looking forward to learn new techniques and apply it in the future. Good morning, everyone. I am Shweta. I'm pursuing masters in zoology from the Department of Zoology itself. And I'm doing specialization in genomics and evolution. That's why I'm here, and I'm looking forward to learn more about this. Thank you. Good morning, everyone. My name is Harsha, and I'm from University of Delhi. And your PhD scholar at Regional Center for Biotechnology. My PhD project mainly revolves around transgenomics and skin pigmentation, and I'm hoping to learn how to analyze big data and predict some fruitful results from my project. Good morning, everyone. My name is Sanju Yadav. I'm pursuing MSc Geology from University of Delhi, from the Department of Geology, and my specialization is in genomics and evolution. Good morning, everyone. I am Aishwarya Moody. I am pursuing my MSc from the Department of Ecology, and my specialization is in genomics and evolution. Good morning, all. I'm Nishita Anuja. I'm pursuing my master's from Department of Zoology, and I'm specializing in genomes and evolution.
Good morning, everyone. My name is Shiva Satija, and I'm pursuing my master's from department itself. And my specialization is in genomics and evolution.
Good morning, everyone. I am Sandy Swami. I'm pursuing my master's from the department itself. I'm a first year student. I'm looking forward to learn. Thank you. Good morning, everyone. My name is Vandana Sharma, and I'm currently pursuing a PhD from the past DRDO, and I'm working in a COVID-19 project. So I'm looking forward to understand the data analysis in a better way. Good morning, everyone. My name is Vashaka, and I'm pursuing my master's from Department of Zoology, University of Delhi, and my specialization is in genomics and evolution. Good morning, everyone. I'm Anamika Gujral. I'm doing my master's from Department of Zoology, and I'm currently pursuing my specialization in genomics and evolution. Good morning, all of you. My name is Achal, and I am pursuing my master's from department itself, and my specialization in genome and evolution. Good morning, everyone. Myself Muskan, and I am pursuing my master's from the department itself, and my specialization is in genomes and evolution. Good morning, everyone. Myself Mohini, and I am pursuing master's from the department itself, and I am currently pursuing my specialization in genomics and evolution. Everyone, I'm Camelia Rosas, and I'm a first year zoology student from the Department of Zoology itself, and I'm pursuing a master's here. And I'm hoping this will be a really nice experience, and I can learn this.
My name is Anand Singh Agarwal. I'm a C final English by specialization in to be here.
My name is Ajit Pradhan. I'm doing masters in this department. And my specialization is entomology, and I'm so interested for data analysis, so I took this program.
Good morning, everybody. My name is Monica Sharma. I'm from BSKC Biodiscovery Lab, and I'm their research trainee as data analysis is a very basic part. So I hope to learn more from this workshop.
Good morning. My name is Rishikesh Kumar, and I'm PhD from Department of Geology. I detect and monitor fish with environmental DNA. So I always play with the DNA sequences, and that's why I'm expecting I will learn a lot today. Thank you.
Yeah. Is it done? Okay. So I request Dr. Pradhan to now take the stage and proceed today's workshop. Yeah, this is it. I think we have left with some of the online participants. There are some online participants are also there. So if the registered participants, I will request. There is a Usha Rani. Hello, is Usha Rani there? Shinjini Sarkar. So can you unmute and then introduce yourself?
Yes, sir. Everyone, I am Shinjini Sarkar. I did
You can
MPharm in
put on your
pharmacology
video also so that we
from.
can see.
Yes, sir. Yes, sir. Definitely.
इसमें जो बोलने वाला है उसका आए ना ठीक है okay
Sir,
yeah
am I visible?
Shinjini is there.
Yeah. Good morning, everyone. I am Shinjini Sarkar. I did MPharm in pharmacology from University of Technology. Currently, I'm doing a PhD from Department of Pharmaceutical Technology, Jadavpur University, and my study is based on pharmacogenomics. So I have some basic knowledge of Sanger sequencing, and I want to know about NGS and its applications.
I think.
क्या वो मिनिमाइज नहीं हो रहा
ठीक है ये तो हो रहा है now we can share na is there anyone is there anyone who wants to introduce himself or herself
Kumari Ila is also there. Is she I think our student Kumari Ila.
Kumari Ila can you unmute and introduce yourself
Okay, Okay, I think we can proceed.
We can proceed. Okay. We can open. We have to share this screen.
No, I think okay, हो रहा है.
Fine.
कुछ आ नहीं रहा है.
ठीक है ना?
What you have to do is don't share the tab. You share the whole screen.
नहीं, I have shared the whole screen.
Huh?
I have shared the whole screen.
No, it was I think tab.
आप
वो tab share हो रहा है. Screen भी है. So that is why you are not seeing anything. तो उससे जाके ना full share screen कर दो तब आएगा उसके.
ठीक है, it's now sharing.
नहीं वो नहीं आएगा. तो share full screen, not tab. Stop sharing. Stop sharing. Now start sharing and full screen. Screen आ रहा है ना? Screen को share करो.
Screen. Okay.
And now both of them. Yeah, it is now coming.
So good morning all. So today I will be talking about next generation sequencing data analysis. So from your introduction, most of the students are basically from genomics and evolution. Okay. So as you are aware that NGS is one of the most recent technique where you can sequence the complete genome of an organism in a single day, in quick time. For example, I will take example of human genome. You can even sequence within 2 days. Okay, you must be aware of whole genome of human. Okay, human genome project. How much time it has taken? It has taken something around 12 years time. Okay, so whereas nowadays within 2 days you can sequence complete human genome due to NGS technology. Okay, in this lecture basically I am going to touch upon what is a bit of what are the NGS technique and how we will be analyzing the NGS data. Okay, mostly I will be focusing upon analysis of NGS data. Then in the after our lecture, then we'll be doing hands-on session basically on analyzing NGS data, basically on RNA-seq technique. Okay, differential gene expression analysis. So the lecture is focused mainly on NGS data analysis in healthcare. So to just briefly telling what is genomics, it is basically the study of genome. Okay. So it is study of the haploid set of an organism's genome is basically genomics. Then a brief history when basically you can see the foundation of genomics is laid in 1953, then followed by then DNA sequencer developed by Sangor and Maxim, then automated DNA sequencer developed by Neurot, and in 1995 the first genome was sequenced, which is of Haemophilus influenza. Okay. Then subsequently, the breakthrough in genome sequencing came when we went towards sequencing of complete human genome. Okay, now what is NGS? So high throughput sequencing describes the modern sequencing technologies, that is Illumina, Ion Torrent, Pathway Sciences, and all technologies developed in this new millennium. There are many sequencing sequencers like Illumina, which is second generation sequencer, and Ion Torrent, that is also second generation sequencer. But when you are going to Pathway Sciences and also Nanopore, they are called as third generation or fourth generation sequencer. So using this sequencing technology, the genome sequencing, whenever you are doing, they are considered as the NGS technology or high throughput sequencing technologies. They are referred as next generation, third generation, or fourth generation sequencing methods. They are very fast and generate data from gigabytes to exabyte. As you must be you will be like when you will sequence complete genome of a human, if you are sequencing 30 times at least, then you will be having at least 150 GB data, because you know that human genome is 3 billion bases. Okay, so then when you are sequencing, okay, then it will be generating 150 GB of data in 30x theoretically. Okay, but to get 30x, probably you might sequence you need to have 250 GB of data. Okay, now once you are sequencing genome, what is the goal? As you can see also, the present access technology can sequence the human genome at a cost as low as $1,000. Okay, instead of 0.5 to 1 billion and over a decade of time is taken for human genome project. Okay, completion of human genome project. Then once you are sequencing a genome, what is the goal? The goal is to identify the precise order of nucleotide bases. Okay, once you are sequencing, what is the meaning of sequencing? It is basically you want to know at position number 1, A is there, or T is there, or C is there, or G is there, right? And what is there in position number 2, what is there in position number 3? That is the main goal of human genome sequencing or any organism's genome sequencing, right? To identify the precise order of nucleotide bases in a genome. Okay. Now, once you have identified precise order of the nucleotide, basically you have constructed the draft genome. And generally, if once you are constructing the draft genome, what you used to do? You used to submit in, say, NCBI. If you first of all you used to submit in NCBI, then you will get accession number. Then you will try to compare whatever genome you have sequenced, how it is different from the existing genome, right? When you are studying about evolution, what you are trying to see, what is the phylogenetic lineage of this particular organism you have sequenced. Okay, so first once you are sequencing, you are trying to identify precise order of the nucleotide bases. Once you have constructed the genome through genome assembly technique, next is to identify the evolutionary relationship. Of course, once you have sequenced the genome, then you would like to annotate it also. You would like to see whether any new gene came up, whether a novel gene is there. For example, you are sequencing say COVID genome. Okay, so for example, you would like to see whether there is any differences in genome. You can see once you have sequenced, like you must have all of us are going through a pandemic, right? So in first phase, some different lineages of coronavirus genome was there. In second phase, some other delta strain came up. Now Omicron is there. What are there? There are differences at genomic level. Okay, once you are sequencing a genome, then you would like to see what are the differences. That is called as variant calling. Okay, the NGS technology we are using to identify those precise differences in genome is called as variant calling. Okay, through variant calling, what we'll find out SNPs, single nucleotide polymorphism, or indels, that is insertions or deletions. You even see that there is one C changes to T. Okay, in the first line in the SNPs, you can see that C changes to T. 1 minute. You can see here. This is the one where C it is there. For example, you have sequenced the second genome. Okay, C changes to T. This is SNP. But in the second one, there is insertion. You can see that GAT is present in the reference genome, whereas in the second genome, GAT is deleted. Okay, that is what called as deletion, insertion or deletion, or in other words, indels. Okay, so this is the basic requirement of a genomics project, that you sequence the draft genome, annotate the genome, identify the differences, identify the evolutionary relationship. Okay, these are the basic things. Now, what else we can do? Now, once we are doing something, then we are getting a little bit more greedy. Okay, can we use this genomics for identifying potential drug targets or biomarker or not? So, for example, one person is having disease, another person is not having disease. Okay, you collect RNA data from the disease individual and the normal individual. Okay, you look, generally you must be doing for a single gene. okay, to see the differences in gene expression. But in genomics, what you can do for all the coding genes of a disease individual and a normal individual, you can take and you can compare. If few genes are dysregulated in disease individual with respect to normal, then definitely those genes are having second role in disease process. Otherwise, why they are dysregulated than the normal ones? Okay. So basically, next one we can perform is differential gene expression analysis. Okay. We can take a set of disease individual or set of normal individuals, and we can compare what is the differences in gene expression level. Okay, and we can correlate those dysregulated genes in disease process. Okay, we will be discussing that in detail in future slides. Okay, you can see here an example here I have given for differential gene expression analysis. In the whole genome, out of 30,000 genes, you can see few genes are in green color. Okay, they are either upregulated or downregulated. Okay, and here is an example of single gene. When in condition 1, condition 2, in condition 2 it is upregulated with respect to condition 1. If it is upregulated in condition 2, it means it is having second role in creating the condition 2. Otherwise, why it is upregulation of that particular gene might be associated with the second phenotype. Okay, so this is what we can study with NGS. So this is in brief the NGS technique. You collect sample, go for library preparation, enrichment of target region, then massive parallel sequencing using sequencer. After that, bioinformatics analysis. Okay. Always remember, when you are going for NGS sequencing methods, or you are giving to any company for NGS sequencing, you are likely to get raw data. The raw data will be in FASTQ format. I will be discussing more about FASTQ format in next slides. So once you are having raw data, you can go for variant calling, that is to identify differences at genome level or gene level. Any SNP is there, or any CNV is there, copy number variations are there, or any indels are there. All those things you can identify in your genome. For example, somebody is working in MTV or drug resistant strain and drug susceptible strain. Okay, so for example, you are working on that. So if a particular mycobacterium tuberculosis strain is drug resistant, there might be some changes in genome. There might be some genomic differences in drug resistant strain with respect to drug susceptible strain. So, for example, you will take 10 samples of susceptible strain, 10 samples of drug resistant strain, and you sequence both. Okay, then you use variant calling. The variant calling method I'm talking about. Use variant calling method to identify the genomic differences. Okay, so for example, you will find some variants in drug resistant strain and some in drug susceptible strain. Your goal should be those variants which are uniquely present in drug resistant strain and absent in susceptible strain. If you can identify such variants, then definitely you can link them to the drug resistant strain phenotype. And probably in disease diagnosis, you can use that. So, for example, a new person comes with TB complaint, you take the sample and go for genome sequencing. And if you are identifying drug resistant strain variants in that, drug resistance means they are not drug resistant to all drug. They might be resistant to one drug and they may not be resistant to other. So that you can use in the clinic that this person is having this particular variant, so don't use drug A, rather you give drug B, so that he will get cured. Even if you take drug A, the patient will not get cured because it is drug resistance to that particular drug. Okay, so basically variant calling can help in that aspect. Now next is RNA-seq. Already I have discussed. Yes, yes, yes.
Example that you gave.
Yeah.
Resistant and the one that is not resistant.
Yes.
What you're saying is that you're saying that we can compare the genomes of the two.
Yes.
And then find the difference.
Okay.
Differences are likely to have to be playing a role.
Yes. Yes.
So just can you give an example like how long is the genome, how big the difference should be? I mean, just some quantitative example.
I think let me tell you. Okay. We are not comparing genome. In NGS, we will be getting reads. I'll be coming to those things in my slides. So which are of length 150 bases only. Okay. So when you will go for variant calling, what will happen? you will get say 10,000 variants for drug resistance trait. Okay, because as you know that in NCBI there is a reference genome already there, right? So you are not comparing directly drug resistance versus drug susceptible. You are comparing drug resistance with NCBI reference. You are comparing drug susceptible with NCBI reference. Okay. Now you identified say 10,000 variants in drug resistance trait.
What variant you mean 10,000 these SNPs?
SNPs. Yes. SNPs or in Delta. Okay. Now these two things you compare in susceptible and resistant. Say your 10,000, your 8,000 for example, and common might be 5,000. You remove those 5,000 common. Okay, there might be 2,000 unique drug resistance traits. Okay, I hope it is clear. So that is what, and of course I will be giving another example in human data in future slides. Okay. Now another thing is metagenomics. So as you know that if we are having like any content, so for example we are having any content, then what we used to do? We used to go for blood test. First of all, we got any infection, we used to go for blood test or stool test or urine test, right? Any infection we are getting. So what happens when we are going for that? We are checking, we are going for a serology test, right? Or CBC or any organism like MTB is there or not, any kit-based test we are doing, right? What happens sometimes we will not get any pathogen present. Okay, it will be negative for everything, but still we are having this, right? Why? Because we are looking for only one organism, or we are looking for at most 5 organisms, or at most 10 organisms. We don't know which virus, which bacteria, what infected us, because probably for that serology test is not done. Metagenomics is an approach where you can see any organism present in your sample, you can detect. Okay, NGS can help in that. So this is what metagenomics, where you can identify various genuses, various species present in a any kind of sample, any like swab, urine, stool, blood, anything. Okay, so when NGS can be very helpful for that, you can go for metagenomics analysis and you can identify that.
So for metagenomics, you mean to say from the fecal sample, etc., we can get a sort of all the organisms that are present in the
Yes, yes. So yeah, yes.
But if genomics available on
Not required.
not available on
NCBA. So in the context of what you want to know, that whether it can identify which organism it is or not. To which organism. That is what the phylogenetic analysis will be doing. Okay.
So basically, you get the whole genome of the organism, or are they broken?
In metagenomics or any NGS technology. So the goal is different. Okay. So when you are going for assembly, you get whole genome. Okay. If you are sequencing for a single organism, you will get whole genome. But when you are going for metagenomics, you will not get whole genome. You will get only marker regions. But recently, there is a new technique called as software metagenomics, where you can get whole genome. But what happens, say for example, you are sequencing human as well as virus. If you are collecting samples from human, it is likely that you will get more of human instead of virus, because human genome will be predominant, right? Okay. So in metagenomics, you can software metagenomics, you can get whole genome. But when you are going for amplicon-based metagenomics, you will not get, only you will get marker genes, 16S RNA, 18S RNA, or ITS regions. Okay. Okay. So another aspect is epigenomics. As you know that all the disease processes are not due to changes in nucleotide or nucleic acid bases. Okay. There are environmental factors also which are playing role in disease process. Two individuals will be having genome constitution similar. Okay, there is no differences, for example, but still one will be having disease, another will not be. It might be due to they are living in two different places. They are in two different environmental conditions. Okay, so there it might be due to DNA methylation, histone modification, or other kind of epigenetic modifications. NGS analysis can be helpful in identifying epigenetic marks in our genome. Okay, those are associated with the disease of interest. So even you can identify the role of epigenetics in disease process based on NGS. And let me tell you, as I mentioned, that it takes hardly a day or two for sequencing. Of course, library preparation takes some time. You take hardly 7 days. Okay, to start a machine, you require library preparation time. Okay, machine cleaning, sequencing, then machine cleaning. So one for one process you require 7 days, but sequencing takes only a day. Okay, so after 7 days you are ready with your report. What is the reason for the disease? Of course, it requires a lot of hardcore analysis. Okay, you need to interpret your data properly. That is what quite important. Now, so this is what again precisely whatever I have shown in last slide. So what are the applications in NGS? Whole genome sequencing, whole exome sequencing. So in whole genome or whole exome sequencing, basically you can generate clinical report for a disease. Nowadays, if you go to hospital, you must be knowing that doctors are prescribing for whole exome sequencing. I'm not sure how many of you are aware, but if you go to hospital, for most of the genetic diseases, they are not going anymore for single genetic test. You must be aware of Mendelian inheritance diseases. Okay, autosomal dominant diseases, autosomal recessive diseases are there, right? There is clear-cut evidences in that. But nowadays, what happens? Doctors are not suggesting only for a single genetic test. Rather, you go for whole exome. Whole exome means you are likely to sequence almost 20,000 genes, or clinical exome where you can sequence up to 5,000 genes, and you can identify the differences. So based on the whole exome data, you can generate a clinical report that if a person is likely to be susceptible to a specific disease or not, that you can. I'll tell you the process workflow in future slides, but I need to say, if you will just give your exome, or if you will give your broad for whole exome or whole genome sequencing, you can find out the differences with respect to the reference genome, and there are databases, well-established databases, which with known phenotypes, like whether you are having any cancer susceptible mutation, whether you are having any genetic disease like thalassemia mutation, any kind of diseases. Okay, you are having susceptibility to, those can be identified and they can be reported as clinical reports. Okay, so the application of whole genome and the whole exome analysis is that you can go for SNP or structural variant discovery to identify phenotype associated with a specific disease to generate a clinical report. Okay, and it is very much in clinical practice in hospitals nowadays. You go to AIMS, you go to PGI, any renowned hospital, if you go today, there is a well-established genomics lab, or even the private labs, if you go for Alpha and all, they are having cancer panel. I think some of you must be working in cancer or something like that also. Okay, so you will find cancer gene panel, this gene panel, that gene panel, many kind of gene panels. Right? Okay, next is whole transcriptomics, where you can go for transcript quantification, differential gene expression, pathway and gene ontology and treatment, and protein-protein interaction analysis. So what you will get? You will get potential drug targets or biomarkers for discovery of gene expression data. Then in metagenomics, as I have mentioned already, you can identify the role of microbiota in various disease conditions to support clinical decision. As I have mentioned, that what happens? Sometimes you will go for test, you will find no pathogen is there, no infection is there. Then definitely you will not take medicine. It is a normal cold fever or viral fever, like that you think, okay, after 3 days you will relax, you will think. But you might be actually having an infection, and there is no serologic test for that, that's why you're not getting as such. Okay, so you will take a broad spectrum antibiotic, you will not take a specific antibiotic, but metagenomics can actually identify the microbiota composition in the sample you have given, such that you can take a actual clinical decision for therapy, clinical decision for taking medicine, specific medicines. I'll be giving you examples also on that. Last thing is that epigenetics. Basically, through epigenetic analysis, you can find out methylome, histone modification analysis you can do to identify epigenetic marks associated with the disease of interest. Next, this is an example of NGS technology and its clinical application in cancer. So again, in genomics, what you will get? Through data analysis, you will get in point mutation, small emails, copy number variations, structural variations. In transcriptomics, you will be getting differential express genes, gene fusion. If two genes are fused, you must be doing fusion gene analysis. Okay. When you are suspecting that there is a gene fusion, you are doing that. But nowadays, there is no just for single gene analysis. Simply go for whole transcriptome and do analysis to identify if two genes are fused together or not. Okay, you can do fusion gene analysis. Alternate splicing, you know that splicing event happens in central dogma of biology. All of you are biologists, right? So you must be knowing that it is from DNA to mRNA to protein. And in a genome like human, there is only 1% coding region or 1 to 1.5% coding region. 98% to 98.5% are introns. And again, in a single gene, all exons are not participating as a transcript. There is an event of splicing. Okay, there might be exon skipping, there might be intron retention. Okay, so one gene might be having 8 transcripts, and all transcripts may not express in a single organ. Different transcripts might be expressing in different organ, right? So that is happening. So that event you can study. You can know that actually which transcript is expressing in liver, which transcript is expressing in kidney, which transcript is expressing in blood. Okay, if you are having a particular disease, if you will be targeting other transcript, then say for example there are 5 transcripts and you and 5th transcript is expressing in liver, you are having a disease in liver, and you are targeting the 6th transcript, you must be knowing that structure-based drug designing. Okay, where a protein is targeted to design drug molecule, and transcript-wise the structure will vary. So if you will target 6th transcript instead of 5th transcript, then disease will not get cured, medicine will not work. Okay, so it is quite important to identify which transcript is present in the disease tissue. Okay, that you can identify. And of course, you can do RNA editing through transcriptome analysis. Our hands-on session today would be majorly focusing on RNA application. Okay, we'll be more talking about differential gene expression analysis. I would suggest all of you to log in with a laptop, probably will be provided, or if you are having. So we'll be taking an example and we'll be performing differential gene expression analysis to identify potential drug targets from the RNA data. Of course, another thing is the epigenomics, where you can study methylome, histone modification, and transcription factor binding. All these things you can actually integrate to for further understanding of cancer. So what happens? So how to use this data in healthcare? Basically, disease diagnosis. You can use this data, NGS data, for disease diagnosis, vaccine design, drug target discovery, and personalized medicine. This is an example, as you can see that that you have sequenced your complete genome. Okay. Now, basically, when you are going to this is the precision therapeutic I am talking about. Okay, you are going to doctor, then you are telling these are the differences in my genome with respect to the reference genome. Okay, then probably there might be 5 medicines, but doctor might prescribe you 1 medicine. Okay, this is what is the precision therapeutic. Okay, we have spoke a lot about what is NGS and what kind of applications it might be having. Yes, please go on.
Sorry, very basic question.
Yeah.
By transcriptomics, you mean to say the genes they are being expressing the right thing or not?
Yes. Yes. What is the quantity of expression of specific transcript?
The quantity of expression.
Yeah. See, for example, one person is cancer, another person is a normal individual who is not in cancer. You take a tumor tissue, RNA, right? And normal tissue RNA.
RNA means the gene.
RNA, yeah. RNA means RNA.
The mRNA or the.
Yeah, it is total RNA and mRNA you are doing. Total. Okay. You can do even long coding RNA and all, but majorly we use to see mRNA. Okay. Okay. So normal tissue RNA and cancer tissue RNA. Now, in a normal individual, what happens? There is a specific level, right? Say for example, you cannot define what is the level of expression. So for that reason, generally what happens? At least 3 normal individuals, 3 cancer individuals. At least I am saying, but it is not the threshold. Okay, you should be taking 15-15, 20-20, or 50-50. There are projects where we used to take 100 plus 100. Okay, so what happens? More samples you will get, more confident you will be on their expression. Now you have taken the normal individual, you can identify the mean expression of the normal individual, right? And you can find the mean expression in the cancer individual. Now once you are comparing, you will know how many fold differences in expression in cancer. It might be upregulated, it might be downregulated. If it is upregulated gene, then you would like to design an inhibitor to control its expression so that cancer will get cured. This is very basic, as I said. It is not like if you are really lucky, you will get one, and you can design a drug molecule and you can cure cancer.
So at the genome level, things can still be same, but the expressions could be different.
Yes, yes, yes. Why I am telling? Because it is not only the mutations leading to differential expression. It might be due to there is change in mRNA expression that is leading to gene expression. Okay, it might be due to the methylome pattern. It might be due to the histone modification. Your expression is different. So genome might be same, expression might be different. Okay. So now, yeah, yes, please.
So what is the main difference pattern of the cancer patients and the normal
Okay. Uh, uh, it means there is no difference, right? Technically, there is no difference, right? Okay. Now, do you believe that it will happen? When I will take one gene, it can happen. When I am taking 30,000 genes, when I am taking 1 lakh transcripts.
So if you're targeting a particular tissue or a particular organ
Yes.
of a cancer patient and a normal patient,
Yes.
and then
Mhm.
and both are
Both are saying is there is no difference that cannot be targeted.
the what if the disease is not dormant at that stage,
Mhm.
but it's still present in the cancer.
That's why you need to go for single cell transcriptomics. Okay. So it might look like because when you are taking tissue, you are taking even almost a normal cells will be coming. There might be experimental error also. Okay, or there might be, as you were told, dormant, or some genes might be expressed in fibroblast cells, some genes might be expressed in immunocells. But when you are taking tissue, you are crushing everything. Okay, and you are comparing RNA. That's why there is a new technique called a single cell transcriptomics, where every cell population you can take. Okay, and you can in norm as a whole that it may look like there is no difference, but in cell population when you look there might be difference. Okay, and it can never happen that there is no difference. If there is differences, probably you have added more normal tissues to that, or probably your sample size is less that there is no significant figure. Okay, so for example, you will take 3 samples. You can't expect that pulse discovery rate will be significant. At that point of time, you should close your eye on statistics. You should just look for fold change. You get my point? Okay. So this is what. So another reason is that then there is no clear-cut biomarker. Generally, it never happens. Okay. But if it happens, then you need to go for single cell technology. You should prefer single cell technology. Okay. Yes, please.
Sir, you explained that we can identify which transcript is expressed in which organ.
Yes.
Sir, can you please elaborate on this?
See, as you are aware that a single gene might contain multiple transcripts. Okay, and always remember all transcripts are not getting expressed together altogether. Okay, so some organ they are called as isoforms, right? So in some organ isoform 1 will be expressed, in some organ isoform 2 will be expressed. Okay, so when you take data from multiple organs, you might find that in one organ 2 isoforms are getting expressed, in another 3 isoforms are getting expressed. Quantity might be different. Okay, quantity might be different. Okay, so when you are identifying a gene is dysregulated, it is quite important to know that which isoform is dysregulated. If you do not know which isoform is dysregulated, then if you go for drug designing by targeted drug designing, your drug design cannot be perfect. You might be designing drug for the same gene, but because of different isoform, there might be slight change in the cavity, binding pocket. So drug designing may not be perfect. Okay. One more thing I would like to tell that when you are performing transcriptome analysis, you might get somewhere 2 isoforms are upregulated and 1 isoform is downregulated. And when you merge everything, normalize everything, you might find no differences. Just coming back to your question. Okay, that also happens when you are doing practicals. Okay, so please never generalize in terms of gene. Always, therefore, transcriptome analysis are not performed in terms of genes. Genes are reported only for our understanding, but actually transcriptome analysis basically performed based on transcripts, probes. If you are going for micro data analysis, you will find probes. If you go for NGS data analysis, you will find transcripts. There might be 1 lakh transcripts, 1.5 lakhs, 1.05 lakhs transcripts. Out of that, how many transcripts are distributed? Later on, how will communicate that to in publication? So for that reason, we are using it in symbol. But again, when you are presenting in a paper, research paper, please try to mention the transcript name. Even if you are downloading data, some of you must be downloading data from NCBA or REPSI. Okay, so you must be knowing there is a transcript ID with respect to that. Okay, they are telling this is the protein, but which transcript it is, that information also used to be there. Okay, so never generalize. When you will go for analysis, always you would like to generalize that, because you want people to, you want your data to be more presentable to people. But that is not the way how you will be doing research. When you are presenting the genes, you should also mention the transcript somewhere, either in supplementary material, or else when people will be following your research paper or your thesis, when they will try to take that work forward for rational drug design, which is the next step, they probably either they have to take all transcripts. Okay, if you are not mentioning, again experimental, like you are spending more money on that. Okay, because you didn't pinpoint it to a specific transcript, so they are going to evaluate all transcripts. That's what. Okay, any other questions? Okay. So now we are coming to some of raw data. Okay, when you are how a NGS raw data looks like. NGS raw data looks like this. Okay, you can see the black screens. More of black screens I will be showing you today. There will not any graphical user interface. Okay, be prepared for that, because when you are doing NGS data analysis, the data size is quite large. You can't keep 1 TB of data in your laptop, right? You cannot work on your laptop by keeping 1 TB of data. Neither your laptop will be having 120 GB RAM. Okay, so you require a server for that, and Genomic Center at ICMR is providing that facility, and you are always welcome to use the facility whenever you require. Okay, if you are having any specific analysis to be performed, you can always write to Genomic Center. At end of the lecture, I'll be showing you one website where you can register your request to utilize Genomic Center facility, because you cannot work in laptop. Okay, but you can access our server through that. Okay, that you will be doing today. So when you are, you must have downloaded data from GenBank, right? NCBA. You must be aware of NCBA. And in NCBA, there is a database called GenBank, which is having the gene sequences. And UniProt is a database for protein sequences. And when you are downloading data, it will be in protein sequences. And which format they are in? Any answer? FASTA format. Perfect. Can anybody answer that? What is FASTA format? Okay. Perfect. So if I am correct, this is the representation of FASTA format. It generally starts with a greater than symbol with a header line. Okay. Then from second line onwards, it is the nucleotide of protein sequences. Right. In GenBank, if you are downloading data, it will be in GenBank format. Right. So there are many sequence formats and all. Okay. So, but generally in all format, you will get a nucleotide sequence and simple header line. What is FASTA format? FASTA plus quality. NGS data is not generated in FASTA format. Rather, the raw NGS data is presented in FASTA format. Okay. What is FASTA format? It means FASTA plus quality. You can see here. I hope it is visible. Here you can see there is a header line, right? At the rate 1/1/1. Is it visible? Okay. So that is header line. So every read in NGS data, we don't say them as anymore as sequences or genes. Rather, we represent them as reads. Why? Because they are of length 50 to 150 bases. Often they are of length 200 bases. Often they are of length 300 bases, but not more than that. Of course, if you go for packbile sequencing, Oxford nanopore sequencing, you might get long list that is more than 100 bases also. Okay, but generally the second generation sequencer like like Illumina platform, you'll be getting short reads that is 100 to 150 bases in general. Okay, so there will be 4 lines for each read. First line is header line, second line is the actual sequence line, third line is a plus symbol that is called as separator line, and fourth line is the quality line. Okay, that's why it is called as first queue format. Quality line, as I say, faster plus quality. That's why it is called as first queue format. So why it is required to represent quality line? Is it necessary? Yes, it is necessary, mainly because NGS is a high throughput technique. Okay, when you are working with high throughput technique, there is every likelihood you are going to get like wrong data also, false positive data also. Okay, therefore for base call, for every base call, if you are telling position number 1 A is there, means what is the confidence of A? That we should mention. It is 90% confidence. If I am telling position number 1 A is there, what is the confidence? What is the accuracy? It is 90%, 95%, 99%, 99.9%. Some accuracy should be there, right? If I am telling 90%, it means out of 100 bases, there might be 10 errors. Then those 10 errors might be variants. All wrong, right? Right? So in NGS data, the very first step is to analyze the raw reads to perform quality control. Such that we are going to take the bases. Those are having accuracy greater than 99.9%. Greater than 99.9%. Okay? That is represented by a value called as thread score. Okay, thread score 30 means base call quality is 99.9%. Okay, thread score 20, base call quality is 99%. Thread score 10, base call quality is 90%. Okay, this is what you need to remember. So basically the last line you are seeing here, that is dash dash AA. You can see the last line. I would rather show in mouse. You can see here. This is A. First position is A. Okay. Then there is a plus symbol. Then there is a dash. Okay. Now you must be wondering what is this dash? I was talking about 90%, 95%, 99.9%. What is the dash? This dash is nothing but a ASCII symbol. In your computer fundamentals, you must have studied that every keyboard character is having a numerical value, right? And that numerical value is called as ASCII characters. Okay. If you look at my next slides, I have mentioned the ASCII symbols. So these are the ASCII symbols. Okay. So ASCII symbols means, so for example, if you will go for dash, what is the ASCII value? It is 45. The ASCII value of dash is 45, right? I pointed my mouse there. Okay. So it means if you will come back, the first character A is having a ASCII value of 45, but it does not mean the quality is 45. Okay. So how the thread score is calculated? Thread score is calculated like 45 minus a offset value, sequence or offset value of 33. That is quality value is 12. That is Q12. I have mentioned Q10 means 90% accuracy. Q12 means 91.8% accuracy. Okay, Q12 means. So Q20 means 99% accuracy. So now 90 to 99. If I am telling it is 12, it is 91.8% accuracy. Okay, so the base call confidence is 91.8% for A. Now going back to the slide again, if you look at the third base, that is T. Okay, look at the mouse pointer again. The third base T, it is having a base call quality of capital A. Okay, what is the ASCII value of capital A? If you will come at capital A, it is 65. 65 minus 33. 65 minus 33 is equal to 32. So it means thread score is Q32. Okay, so again it is more than 99.9% accuracy, right? So our goal should be 65 minus 33, it is going to be 32. Okay, so our goal should be we should be selecting reads, because as I mentioned that when you're going for FGS sequencing for whole genome of human, there will be 150 GB data. Remember, 150 GB data means how much data you cannot open in your Windows machine itself. Double reading you cannot open. Okay, you have to use Linux machine to use the command called less, which I will be teaching today to you again. Okay, you can open that, but then it is huge data. 150 GB when you are studying how many video files you can be storing in your laptop. Okay, so it is huge data in fact. Okay, so we cannot manually screen them. Huh? We cannot manually screen them. So we need to use computer program to screen those leads or those bases are having base call quality more than 30. If you are working on Illumina platform raw reads. Okay, the condition is that if you're working on Illumina platform raw reads, then you should go for Q30 reads. Okay, because Illumina generates high quality raw reads. But provided you are working on iron current data, iron current is another sequencer. Okay, iron current data, then you will never get Q30 reads. Okay, you will never get Q30 reads. Mostly reads will be Q25. Means the accuracy is 99%. Machine accuracy is like that. So you should be keeping a threshold of Q20. Regular point. So if you are analyzing, so for example, today I will be teaching with you linear data, tomorrow you are analyzing handwritten data. You will press a threshold of Q30, all your data will go. Okay, so the quality, of course, we know that higher the quality, better is the confidence. But then, if you are losing all your data, then what you will analyze? So you should know what is the threshold you are giving, what is the error you are allowing. When you are taking your data with 99% accuracy, you are telling, you know yourself that there is one false positive base in 100 base calls, right? In every likelihood, there will be one false positive base call in 100 bases. You know that, okay? So if your variant frequency is equal to the random frequency, then all value false positive. Your variant identification when you are doing just be more than the random frequency. Of course, there are other issues while variant calling. I'll be discussing. Yes, please.
What are the factors that affect?
Okay. Factors, see, it is a pretty automated process. Okay. Confidence on base call is basically dependent upon the reagents you are using. So generally what happens when the quality will decrease when your reagents are decreasing. Machine sequencing is ongoing, very last steps. Okay, reagents will be decreasing, so sequencing will not be okay. If there are contaminants, in your say for example you are taking DNA sequences, but there are contaminants. Okay, it is likely to in every likelihood you will be getting these issues. Another thing is that the beginning of the bases in a read, beginning reads and end reads, they are also likely to be of poor quality. Okay, so that's what. And I would say rather, see, it is basically a machine things, automated. Now their things are very automated. Okay, once you are feeding, if your library preparation is perfect, there won't be any issue in while sequence. So major aspect is your like then what happens in RNA? RNA is quite degradable, right? It will get degraded. So quickly you need to place in RNA letter and you need to collect it from surgery room, for example. Then press in RNA letter and you take it to your like library preparation. Okay. So what happens if your library preparation is perfect? In library preparation enrichment is perfect, it means there won't be any issues while sequencing. Only issues will be when reagent will be completed, at that point of time, you will be getting good quality reads. But then that's why we are using bioinformatics approach to clean those things. We'll be keeping only good quality, we'll clean the good quality so that your results will not be getting false positive. You might be losing something good things also. But then your results, whatever results you will be having, will be true positive results. More of true positive results. Okay? Yes, please go on. It is actually a sequencer offset value. Okay, because sequencer is assigned the quality value. Okay, so there are two kinds of offset values sequencers assigned. Different sequencer like Solexa platform, they are having offset value of 64. So what I have, because thread code is a manual. For example, you are using, for example, you could have used one also. Directly they could have used, but then they find if the ASCII symbols are more relevant to give from that number. Okay, so when in matching the head, whatever number they have given, they mention it is offset value of 33 into minus. Okay, so if it is set in sequencer, you have to follow that. Any other questions? Okay. So as I mentioned that you should be preferring Q30 reads, okay, when you are going for NGS sequencing, if you are working on Illumina platform. And so now we are pretty clear about what is reads and what is the quality values. Everything you will be doing in practical. Yes. Now coming to analysis, I will be giving a brief overview of analysis. What kind of analysis you can do with NGS data. First step of NGS, once you are having this, first step is quality control. Any kind of NGS analysis you do, any kind, like you do RNA-seq, you do variant calling, you do metagenomics or epigenomics. Any analysis you do, first step should be quality control. Okay, you can go for quality control, and if you are going for variant calling, if your goal of your project is to identify genetic variants, then you can go for read mapping. Reference genome is there in NCBI. Collect the reference genome, go for read mapping. Now, once you have mapped the read, then go for depth and coverage analysis. Why you will be going for depth and coverage analysis? For example, you have given it for sequencing to a service provider, to a company. Okay, yeah, you are told that okay, we'll be giving you 10 GB of data at 100x sequencing. What is the meaning of 100x sequencing? Means every position in the genome is sequenced 100 times. Yes, every positions in the genome sequenced 100 times. That's what you are going to do, right? So how you will check? You need to check, right? They have sequenced, they have given 10 GB data. 10 GB data might be in contaminants, right? Might be in poor quality reads, might be in adapters, sequencing adapters. So you need to check whether whatever they committed, that 10 GB data at 100x with 95% of genome coverage. All the terminologies we will be discussing in detail, but this analysis you need to do. Okay, that is called as coverage analysis. Then variant calling, variant annotation. You have identified a variant, for example. The next thing is that you need to annotate it. This variant is part of which gene? Okay, it is present in exon region or intron region. It is then next thing is that it is present in promoter site or any other site. So all those things you need to look at. It is a synonymous variant or non-synonymous variant, right? It is having an impact on gene function or not. So all those things you need to annotate. Of course, that practical we will not be doing today because one day is not sufficient for all those things. But that is what is the goal of variant calling. Then after that, what is the result? What is the outcome? Case, clinical report generation, case control analysis. For example, you want to say, for example, you want to study in discovery mode. You want to discover something. Like clinical report means whatever is discovered, you are using that information to tell that okay, this person is having disease or not. Okay, right? But when you are working in discovery mode, you want to identify new variants which is associated with the phenotype. Okay, so that you can do. So case control analysis, familial analysis. Okay, all those things you can do. If you are going for de novo assembly, so as you can see, if you are going for de novo assembly, as I have told that most of the sequencing projects focus upon genome sequencing, whole genome, draft genome construction. So if you are going for de novo assembly, go for QC, scaffolding, merging, ordering, then draft genome construction. Then once you are constructing draft genome, the goal has to be to identify potential drug target. Say for example, you have sequenced MTB genome. Okay, one goal is to identify the variants present and associated with the disease of interest, right? Next goal should be to identify a drug target. Okay, a gene which is essential for survival of mycobacterial tuberculosis and absent in host. Okay, so if you can identify such genes, then you can design a drug molecule to block function of that gene, that essential gene, or you can knock out that gene. Okay, if you can knock out that gene, the pathogen will die. Or if you can design an inhibitor drug molecule to block the function of that gene, then the pathogen will die. Okay, drug target identification is going to be the next goal, genome. So that is the application. Drug target identification, subunit vaccine design, and virulence gene detection. Of course, there is a software in TID we have developed, okay, through Bioinformatics Center project, okay, that is available online. If you will feed a complete genome to that software, in half an hour it will tell you what are the potential drug targets. I'll show you that in future, in future slides. In RNA-seq, of course, this is the protocol we are going to study today. Complete practical will be doing, that is alignment and transcript quantification, differential express gene identification, then pathway and gene ontology enrichment and network analysis you will be doing to identify potential drug targets. Now, drug targets for pathogen you can identify through genome assembly and TID, as I have told. But drug target for non-communicable diseases like cancer, there is nothing like external individual, right? It is our body, it is a lifestyle disease. So you have to do differential gene expression analysis to identify in which condition which genes are getting differentially expressed, so that if you can design a drug molecule, okay, to regulate those expressions, so that the disease process will get control. Okay. Now, metagenomics, you can identify representative sequences with taxonomic assignment, and pathway and cluster orthologous group assignment you can do. Okay, so what are the softwares you would be using for this? So these are the softwares for variant calling. I'm not naming everything, but generally GATK, Genome Analysis Toolkit, is widely used for variant calling. And clinical report, as I have mentioned, how it is being generated, it is generated using ClinVar. Okay, ClinVar is a database where what people are doing basically globally, whoever is doing targeted gene sequencing also, if they have identified a variant which is associated with a disease experimentally, not prediction, okay, experimentally, then they used to submit that data to ClinVar, NCBI ClinVar. Okay, so that data can be useful for generating a clinical report. And every diagnostic labs, you talk about Lalpath, you talk about SRL diagnostic, you talk about any Max Healthcare, anything, okay, they are using in background this ClinVar, or there is a database called FGMD, that is a commercial one. Okay, they use these two databases in background to identify the variants for clinical reporting. Now, for de novo assembly, you will be using SPACE, Abacus, Proca, Augustus. Okay, Augustus for gene annotation. Okay, basically, if you must be having gene finding class, you must be reading this Proca and Augustus. Okay, these are the two genome annotation tools, Proca and Augustus. Then TID for target identification, Artemis for visualization. Okay, then RNA-seq, HiSA2, string type, background, Lima, cluster profiler, cytoscape. All these things we will be doing today in practical RNA-seq. Then metagenomics, you can study KIND or KIND2 and HiCROST. Okay, so this is how a clinical report looks like. Generally, when you will go for exome sequencing, you will get a gene. I have given knowingly ABCD gene name, but actually there is a gene name here. Okay, so ABCD I have given means any gene name. Then which chromosome it is present, chromosome location, RSID. Then what is the variant, whether any change in amino acids or not. And please remember, if you know this, you will get hired in any diagnostic company nowadays. Okay, if you know this clinical reporting, any diagnostic lab, not only company, diagnostic lab or research centers. Okay, so but important thing is that you should able to precisely identify the genetic variants which is associated with a clinical phenotype. What important. Okay, your analysis should be pinpoint. Now, whether it is a heterozygous variant, then domain name, of course, domain name I didn't return knowingly. Then it is a likely pathogenic variant. Okay, if a variant is pathogenic or not. Okay, when you are reporting a variant, you should know that the variant is pathogenic or likely pathogenic or benign. When I'm saying there is a guideline called as there is a guideline actually based on that basically this pathogenicity is decided actually. Okay, so if a variant is pathogenic or not, that you have to identify. If a variant is pathogenic, you should be reporting that. Okay, if a variant is likely pathogenic, you should be reporting that. If a variant is benign, you need to reject that. When I am saying a variant is benign, it does not mean that it is nothing to do with tumor and all. Okay, if a variant is benign, it means it is definitely not having any role in the disease process, as simple as that. Okay, so that you need to identify. then you can see that this particular variant is associated with colorectal cancer. Means when a doctor prescribed whole exome sequencing, they have given tissue samples to me. We have sequenced that. We have identified variants. Okay. After that, we have mapped with ClinVar database, as I mentioned, NCBA ClinVar database. Then we have got a specific variant which is associated with colorectal cancer, and I have reported to doctor. Now, doctor is the right person because they know the phenotype also, right? When a person came for complaining a disease, doctor know what is the phenotype. Okay, if this phenotype matches with that, then they can understand, okay, this is the reason you are having colorectal cancer. Now, this colorectal cancer mutation is a germline mutation or somatic mutation, then this has to be answered. If it is a germline mutation, it might be going to the kids also, offspring also, right? If it is a somatic mutation, then it is fine. Okay, so that has to be decided. If it is germline mutation, then probably again you need to check in the programs also. Okay, because although they are not having colorectal cancer today, probably they might be having risk of developing colorectal cancer. Okay, as you are aware, most of the cancer can be treated if they are identified at the stage 1. Because diagnosis is performed at a later stage, it is quite difficult to treat them, or you want to go for chemotherapy, right? So if before the disease only you can identify the risk, then probably you can go for periodical testing, and probably cancer will not go to the 3rd stage or so, right? 2nd stage or 3rd stage. So this is what the prognostic value of whole exome sequencing. Okay, and it is being done at Genomic Center. Let me tell you, the pipeline we have developed at Genomic Center, you need not do anything. You need to just give the exome samples. It will generate a report, something like this. Okay, it is being done, and for using, we always welcome that if you want to utilize the facility of Genomic Center. Okay, now when you are going for whole genome sequencing, you will get a genome like this, and you can go for annotation. How much time I will be having? After how much time? I can take another 15 minutes, right? Yeah. So if you are going for whole genome, then you will get a whole genome like this, and you see these are the different genes, different color we have shown. Okay, if you are going for transcriptome analysis, basically you will be getting differential expressed in green color. There will be a heat map to show that which genes, how they are expressing in different like samples. And again, what happens when you are going for transcriptome analysis, you will be getting dysregulated genes. Okay, it might happen that you might get 5,000 dysregulated genes. Now again, I cannot tell somebody that go and validate 5,000 genes, right? I cannot tell 5,000 genes are drop targets. Okay, some of the genes might be the effect of the disease than the cause of the disease. I need to identify which genes are cause of the disease. That is what is important. Now, technically, if you look at the transcriptome analysis papers, research papers, you will find, first of all, people used to report the top 10 or top 10 down. This is pretty simple way. But all the top 10 are highly overexpressed or highly downregulated. They might be, I'm not saying that they may not be having low. They may be having low, but some other gene which is at borderline might be inducing all these 20 genes to express this much higher. And more often than not, you will find these top dysregulated genes might be common in all diseases. Then they cannot be biomarker. If they are expressed, overexpressed in all diseases, how they can be biomarker or drug target, right? So if you are trying to tell a disease is, so for example, a gene is a biomarker or expression of a gene is a biomarker, it has to be unique to a disease, right? It has to be unique to a disease, uniquely expressed, upregulated or downregulated in the disease process, then only you can say that. Okay, or even if you are telling drop target. So that is the simple way of presenting the top or top down. But the next thing is that you can go for pathway enrichment, gene ontology enrichment and pathway enrichment, such that you can identify which pathways are getting enriched by these dysregulated genes. Now, when you are going for pathway analysis, you know that which pathways are likely to cause cancer, right? And which pathways may not be having even minute role in cancer. Pretty much likely, there are enough literature in the domain, public domain, right? And also you can make your own interpretation. Here, the interpretation of a biologist comes. Here, there is no role for bioinformatics, please remember. Bioinformatician can give you what are the likely pathways. 5,000 genes, if you will identify pathway, one by one, if you will do, it will take 6 months time. for you easily. And again, to compile them, that okay, 5 genes are from a particular pathway, 10 genes from another pathway, it will take another 6 months time. 1 year you will take for only annotation. But a bioinformatician can do annotation within 5 minutes for 5,000 genes. Now he has done your 1 year's work in 5 minutes. It is your work to spend that 1 year in interpreting the things, to go back to bioinformation saying these are the out of 100 pathways you are given. I believe these 10 pathways, based on my scientific logic, based on literature analysis, these 10 pathways are more relevant. Then again, bioinformation can generate a picture within 5 minutes or 1 day for you. That okay, these are the 10 pathways. Then more in-depth analysis he can again. But the role of biologist is quite important here. Please remember, role of clinician is quite important. You have to, for interpretation, you have to apply your skill. You can, if you will ask me, you tell that what are the top genes. You tell that what are the top pathways. Then I will tell based on statistics only, right? If I tell you top 10 genes, you may not exceed the all 100 pathways, right? Then you will lose your data. Okay, that is what quite important. Okay, so this is the image you are seeing. This is of pathway analysis. Okay, now there is a standard recommendation of basically molecular pathology and College of American Pathologists. Okay, AMP and CAP guideline. This guideline is generally followed by any laboratory you are giving for testing, exome testing. Say for example, you are giving to MedGenome, most of the people give to MedGenome, or Supratech, or any company you are giving for sequencing. If you are asking them a clinical report, they have to follow this guideline. Okay, College of American Pathologists CAP guideline, they have to follow. So this is the guideline for analysis. So guideline means you cannot like skip any of the steps. You have to follow all these steps to identify variant, disease causing variant. That is, you are having FASTQ files, as you are aware, they are the raw files, FASTQ, analysis ready FASTQ. Then you align them. I was about to tell you that file formats. Okay, so these are the file formats. FASTQ files. Then when you align with reference genome, here you can see the reference genome. So for example, you are working on human data, then the reference genome is there, right? So reference genome, then alignment. Then you will get, it is not like alignment file, it is a sequence alignment map file. It is not called as alignment, rather it is called as mapping. Okay, when you are using BLAST, NCBI BLAST, okay, you are doing local alignment, right? But local alignment you are doing. When you are using Needleman-Wunsch algorithm, right? I'm not sure how many of you are aware, but you are going for global alignment. Okay, or Smith-Waterman algorithm for local alignment. Okay, you are doing that. But here, there is no real sequence. There are reads, small fragments. So that's why it is called as mapping. Okay, so basically, after alignment, you're getting sequence alignment map file. Then, generally, if you are having 10 GB of data, you will get 50 GB of alignment map file. Okay, then again, if you are having one sample of 150 GB, you require 1 terabyte for analysis, right? SAM file. So SAM file you cannot keep. If you will keep, then definitely it will take all of your space. So generally, the SAM file is getting converted to, we generally used to convert them to BAM file, that is binary alignment map file. Okay. Then after that, we have to process. Then and we have to apply quality control at every level. At raw read level, I have told you Fred score. BAM QC, that is coverage analysis I was talking about. Then variant QC, once you have identified variant, you have to tell that whether the variant is good enough or not. That also you need to do. Okay. Now you can ask me, okay, you have spoke about all this data. Now all this data generation requires lakhs of money, crores of money. How will do research? Okay, it means you should be having funding for that, right? Without that you cannot do. But then you need not worry about that also. This is the place, NGS data resource for bioinformatics analysis. If you know bioinformatics analysis, globally, whoever is working on cancer, dengue, COVID, all the NGS data are being deposited in all these 3 websites. Okay, say for example, you want to work on colorectal cancer. Just type your colorectal cancer in the box I have highlighted. Okay, then you will get all the NGS data present in colorectal cancer worldwide. Whoever submitted, it is present here. Now you go each file and download the first few raw reads and do analysis and plan your research. And you give time in validation of your result instead of starting from NGS. Because there are a lot of data already available in open source and that is freely available. If you want to practice, you can do that. Okay, so these are the source. You can just give SRR ID here, or you can give the name of the disease. Anything you want, you can just give here and you can get the information. Okay, what you require for analysis? Minimum 4-core processor, 8 GB RAM in a laptop. Okay, but having said that, you cannot do variant calling in laptop. You need to use server. This is the minimum requirement of your laptop, which can be connected to server. Okay, where all the pipelines are there. And today we will be doing that. Okay, if you are having Windows system, you need to install Windows CPL Putty to work with the server. Okay, and some of the minimum things you can do in your laptop also, that we'll be telling you in due course. So this is how a quality control report looks like. Okay, basically, as I mentioned, you should be having Q30 reads. Of course, you can download data also using SRA toolkit. It is not necessary that you will go and click and download, because in a server you will not be having any graphical user interface. You will be having a black screen only. So in the terminal, you need to give a command only, right? So you can use the command first to dump to in fact download data, anybody's data, any project's data you can download. Such that, because what happens, you cannot keep your laptop open for 48 hours, right? Okay, so what you can do, but server generally stays open 24/7, right? 365 days in general. Of course, sometimes for maintenance it has to be rebooted, but generally it is 24/7 server stays open, right? So you keep it. So for example, you have to download 100 samples. It might take 2 days time. So you just write a program and keep it for download. After 2 days, come back and you see your data is there. And again, the pipeline we have developed. If you have to do RNA-seq analysis, simply give the data folder name, click again. After 2 days, come back and see your draw targets are ready. Okay, now what is your job? Everything computer done. Then what you have done? You have to interpret. Your job is to apply your biology skill in shortlisting out of 5,000, whatever top ranked genes or top ranked pathways I have given. You apply your biology skill to tell why this set of genes should be targets, why this set of genes should not be targets. Then again come back to bioinformatician. Then bioinformatician will tell you why you are right or wrong. Okay, that's how we can progress to identify potential drug targets. Okay, now having said that, this is the quality control report. And next, this is what I was talking about coverage. For example, this is the reference genome, and these are the 10 reads, and you can see at 10 times it is sequenced, so that is 10x. Okay, the total genome coverage is 90%, genome fraction coverage is 90%, and sequencing depth is 10. Okay, then next is for variant calling. You can see that this particular variant is present in 8 reads. It means in 80% of the reads it is present. So it will be a homozygous variant or a heterozygous variant. We are deployed in nature, right? Yes or no? We are deployed. Human genome is deployed. So it might happen that if a variant is heterozygous, you are telling, it means it should be present less than 50% of the reads. Technically, if something is present more than 50% of the read, it is a homozygous variant. As simple as that. Homozygous variants are more likely coming from the parents. Heterozygous might be somatic, may not be somatic, because often it might come from a single parent. Okay, that has to be validated later on. So these are the criteria how you will validate various variants, if a variant is of good quality or not. Like one of the example only I will give you. So for example, you have identified a variant which is associated with cancer. Okay, you have identified 5 variants. Now, how do you think that whether this variant is disease causing or not? Of course, there are many criteria. But if a variant is present in high frequency in normal population, in 20% of the population, even if it is pathogenic, it is showing, even if it is non-synonymous, okay, then also it cannot be disease causing. Because if it is present in healthy individuals, 20% of the healthy individuals globally, it cannot be disease causing. Otherwise, why all 20% people are not getting disease? Okay, so you have to use one of the major criteria in screening variant is to set up minor allele frequency. You should select those variants those are having minor allele frequency less than 1%. It means they are extremely rare in normal population. That is, they are existing in less than 1% of the normal population. Okay, this is one of the filtering criteria. of variants, raw variants. Of course, there are databases like Linbar, Cosmic, FarmGKB to actually annotate. So this is a standard VCF file. I'm not going to tell in detail. For damage score prediction, basically you will be using SIFT and polyfan, intervar, snippets. Basically, this is how from a single genome, basically you will be getting 2 lakhs variant. From 2 lakhs to 100 variants, how you can shortlist. Okay. So coming to transcriptomics, these are the steps. We will be talking more in detail in practical session. But to shortly telling, it is basically QC you do in first QC and chemomatic, and alignment do in HiSAT2, and transcript quantification we will be doing in string tag. Differential expression analysis we will be doing using Lima, and then of course in R session, like Dr. Gitya would be taking shortly, there she would be talking about R. So Lima is basically a bioconductor package. So later on we'll be using that. Okay, once you are identifying the different distributed genes, you can go for gene ontology enrichment, pathway analysis, and functional module analysis. This is what the results looks like. You will be generating a heat map. You can see that there are 2 different clusters, case and control. Based on transcriptome data only, you can identify this. And volcanoplots, as I have mentioned, they are showing the distributed genes. So this is the pathway map. It looks like once you are generating, you have got 2000 distributed genes, you can just get pathways something like this. One important thing I have not told is network analysis. So one thing is that top distributed genes. Second thing is that pathway enrichment. Third thing is network analysis. As I mentioned, to identify cause of the disease, there are many genes. So for example, let me give you one example that there are 100 distributed genes. If one gene out of the 100 is connected to 50 genes, 50 other distributed genes, then that is going to be more important because it is having more connections. If I design a drug molecule by targeting that, all the other 50 diseases also will get affected. So that will know based on network analysis. That is called as hop gene identification. This is the research paper you can follow by taking open source data. We have in fact analyzed, okay, and we have identified hop genes. So again, network, as I have mentioned, biomarker, there is a database called as DisGenNet, where you can actually identify which genes are associated with single disease. Okay, as I mentioned that if a gene is associated with multiple disease, generally what happens, it cannot be a biomarker as such, right? Because it is present everywhere. Then how you can say that it is biomarker? It has to be unique, right? So for checking uniqueness, you can just use a database called DisGenNet. Okay, so then you can just generate a network like this, and you can know that which genes are unique. So that's what in genome assembly, these are the steps already I have discussed. Okay, I'm just skipping this, but I would just cover one thing, that which kind of technique you should be following for what kind of data. As I've told that there are many kind of NGS data, Illumina, Ion Current, then PathBio, all those data. So for example, you are having Illumina or 10x Chromium data, then you should be preferring these softwares. Whereas if you are having long read sequencing data, you should be preferring these softwares. Okay, and if you are having both kind of data, then you should be preferring Masurka. Okay, these are the softwares you should be preferring. Okay, then followed by, then you can evaluate your genome assembly for its completeness by using N50 statistics and then genome fraction coverage. And next is annotation. Annotation you can use these are the tools. You can go for gene finding using ab initio method, evidence-based method. Okay, and you can use BLAST, BLAST2Go, GoFit, Amigo, Eggnog, Proca, Augustus. So all the softwares you can use for annotation. This is the software PID I was talking about, where you can give genome as input to identify potential drug target from whole genome sequences. Okay, once you have annotated the genome, you will be getting protein sequences. Give input to this software. It will give you in half an hour to 2 hours. It will tell you what are the potential drug targets, how many virulent genes are there. All those information it can give you. Now metagenomics already we have discussed, so I'm skipping these slides, but just I'll be telling you 2 types of metagenomics. One is amplicon based metagenomics, another is shotgun metagenomics. Okay, so in this, the beauty of this approach is that you can find out any organism, any infection present in your sample, irrespective of it is evolved or not. Okay, that is, and remember, exome is in clinical practice, metagenome is not. Mainly because there are challenges. Once you are getting hundreds of genomes, you never know they are contaminants or real organisms. It might be experimental contamination also. Okay, so that is a challenge. So the bioinformatics protocol is still yet to be standardized. Of course, there are protocols, but it is yet to be standardized for implementation in clinical care. So this is one example. I would say how probiotic you can develop. So this is a patient. You can see the bacterial composition, microbiota composition of patient, and this is of healthy individual. You see here only you can see 4 bands, but here you can see multiple bands, right? If you can enrich the harvest the stool of this patient, and if you can develop artificial stool, okay, from there you can develop probiotics, such that this disease individual can be given the probiotic. Probiotic everybody nowadays we are taking, right? Okay, probiotic we are taking for gut microbes, healthy gut. Okay, so probiotic you can develop such that this can be rectified. Synthetic stool you can develop, simply you can develop, and or you take sperm from their synthetic the microbiome composition, you can develop a probiotic, and it can be given to patient. Of course, all these are theoretical aspects. Yes, please go on. No. Actually, it is talking about different genus, different species. Okay. Yeah, those are samples. Okay, columns are basically different organisms. Okay, so that's what this is a paper from. This is collected from a paper, Nature Reviews Genetics. You can just follow this. Okay, this is quite useful application in clinic actually. Okay, and also you can do transcriptome analysis in infection state and non-infection state, which genes are dysregulated. This is how a standard way of utilizing metagenomics based NGS in clinical setting. Okay, I'm not going into details of this, but I would tell you, you can ask me that you are talking a lot of about NGS things implemented in clinical care. Okay, exome you told that yes, it is implemented in hospital, but whether metagenome is successful as of now, already I have told that it is not in clinic, but in sporadic cases they have been used. Here is a successful example where in a 14-year-old critically ill boy with meningoencephalitis, neuroleptospirosis was detected through metagenome sequencing, and such that targeted therapy was made and person got cured. If neuroleptospirosis was not detected, then person would be dying, because as you know that leptospirosis is a zoonotic infection. Generally, it is coming from the pet animals. Okay, or sanitizing activity people are involved, or soldiers when they are going for rescue operation, they are getting infected with leptospirosis, and it is leading to jaundice. And generally, severe cases of leptospirosis can be fatal. Okay, so once through metagenomics, and once some person is having meningoencephalitis, you cannot suspect that that person will be having leptospirosis also. Okay, but after metagenome sequencing, it was observed, then targeted therapy was given such that the person can be cured. Okay, there are many tests are also available, that is for multiplex pathogen detection, culture negative sepsis, endocarditis, febrile neutropenia, fever of unknown origin. All these things you can, there are kits are available, you can use NGS. So metagenomics based NGS can be key driver in precision diagnosis of infectious disease to advance the precision medicine effort in the healthcare sector. What are the challenges? As I mentioned, it is having challenges, like you never know that it is a contaminant or it is a real microbe. Okay, only you should have enough skill to differentiate that, point number one, and that comes with practice. I cannot say that verbally that it is a really contaminant or not. Once you are doing enough analysis, you will get an idea. These are likely to be contaminant and these are likely not contaminants. Next thing is that doctors has to be also aware about this, and they should be positive to use this. Okay, and next thing is that as you know that standard protocol for developing a clinical decision support system based NGS is yet a work in progress, unlike whole exome sequencing. Okay, so that's all. In the end of my lecture, I will just tell you one thing. This is only for your learning. This one we are not going to tell in practical. This is a server, Galaxy server. It is a very good starting point for learning NGS. At least it will give you idea that what are the softwares are there for analysis of NGS data. RNA-seq, variant calling, metagenomics, you will get it. Why, you can ask me why you are not saying that to use this. You can use this, but uploading 100 GB of data is not that easy. For 2 GB data upload, there will be multiple times it will be breaking, right? So it is not that easy. Point number one. Point number two, when you are analyzing with this, okay, you don't have that much flexibility. Okay, first is uploading. Next, point number three is that if you are working with your original data, you won't like it to upload to somebody, give it to somebody. Rather, you would like to work in your server, which is confined to you, because you have spent crores of money and you would give to some foreign country that server that you take out data. Before you are publishing, they will publish. You never know. Okay, you never know. So what happens? Nothing against it. This is a very good starting point, but I would suggest if you're working with original data, work with command line, Linux. It will be pretty faster, and it is once you will get used to, it will be quite useful also for you. Okay, so that's all. You can create an account in Galaxy. It is a very good starting point. In the event, I also learned NGS by registering to Galaxy. So I would suggest please register to Galaxy. You will get an idea what are the softwares you should be working on. Thank you. That's all. Let's see you again in hands-on session. Okay. Any questions?
Anyone can ask any question if you have.
Yes, please go on. Yeah. Okay, so that is the practical will be doing today. So I think that will be more clear there, because here whatever I will be telling you kind of haphazard. Okay, but having said that, once you are having a set of genes, disordered genes, if you will feed to a software, it will give you what are the pathways they are getting engaged. In fact, in my laptop, the data is there. When I will show you practical, I will show you that. And all of you can install Cytoscape in the computer. Laptop, if you have brought, also Cytoscape. There I teach you network analysis. So today, all these things practically to be done. Okay? At that time, I will clarify. Any other questions? So if no questions, thank you. See you at again during practical.
Thank you, Dr. Pradhan, for a very informative talk. And now I welcome Dr. Geetika to give a talk on for data sciences. She is from ICMR East Genomics Center. I welcome.
और यहाँ पे इंटरनेट है ना इसमें चार्जिंग पे लगाना चाहिए पड़ सकता है आप चार्जिंग पे लगा दीजिए नहीं ये स्क्रू और राइट क्लिक ही नहीं हो रहा Mic. ये यहाँ पे वर्क करेगा और ऑन और ऑफ कर दीजिए ऑन।
Screen share.
स्टार्ट कर। ये 30 मिनट वेट चल रहा है ना।